Meiosis is similar to mitosis because it's about cell division and it follows interphase. So you see here interphase and then starts meiosis. The difference is a couple of things. We already learned some of the differences. So where does meiosis happen in your body? What types of cells? Sex cells, okay? It does this to prepare for what? Reproduction, or to form a new organism. So your cells, your sex cells replicate in order to eventually become fertilized. That's their focal point, let's say. How many cells are made during meiosis? Four. Good. And what do they look like in comparison to the parent cell?
Different.
How? Not combination. Remember, they're preparing. Good. It's half of the parent cell. So the genetic material is half of the parent cell. If you look at this first cell in interphase, there was 4 chromosomes. All the daughter cells have 2. So it's half the number of chromosomes. What else can you tell me about the daughter cells that are different from the parent cells? Can you look at that really quick? You mentioned different colors. What's happening is they're exchanging genetic material. So the genetic material kind of gets mixed. So there is some mixture there. Besides half the number of chromosomes, there's mixture. And I'm going to explain that in a minute. Meiosis 1 is similar to, well, meiosis has 2 parts. I should say that. Meiosis 1 and meiosis 2. So let's go back here and say that. It has 2 parts. Oh, I can't type with these. Meiosis 1 and meiosis 2. Meiosis 1, it'll say prophase 1, metaphase 1, anaphase 1, telophase 1. It just adds a 1. And it's very similar to mitosis with a couple of differences. Similar, I mean, during prophase, the nuclear envelope disappears. The nucleus disappears. The chromosomes become visible. The difference is that chromosomes line up in 4s, and they call that a tetrad. So if you look here, they're lined up. So this would be the 4. 1, 2, 3, 4. So those 4 sticks lining up together of pieces of chromosomes. They're side by side, and they call that they're next to their homologous partner, which is basically same looking chromosome. When you study genetics and karyotyping and chromosomes, you'll see that each chromosome has distinctive functions or genes on it. That's what they mean by lining up. So they're lining up to their partner that looks similar. And then this thing called crossing over starts to happen. And if you look at the picture there in your notes, you see how the blue crossed with the red? That's what crossing over is, okay? I also pointed to it right here because there's another crossing over taking place right there. That's crossing over. If you need me to zoom in so I could show you better, right here you've got some crossing over. They're actually circling it. And here, there's some more, okay? Now, I don't know where I stopped. Let me fix it. Now, as a refresher, besides the nuclear envelope disappearing and the chromosomes becoming visible, what else happens during prophase? The spindle forms. And what else? Not the chromosomes. The centrioles move apart. So you got nuclear envelope disappearing or nucleus disappearing, chromosomes becoming visible, but they're in 4s, which is a tetrad, with their homologous partner. The spindle appears and the centrioles move apart. Then we go to metaphase. What happens in metaphase? Say the 2 things. Good. M for metaphase, M for the chromosomes line up in the middle. Okay, what else happens? That has to do with middle. Good. The spindle attaches to the centromere. Why is the spindle attaching to the centromere?
So it can align everything right.
So it can align everything in the center and do what? Next. Pull it. Okay, that's what it's doing. Now, when the chromosomes line up in the middle, how are they lining up? In fours again. They're lining up as a tetrad or as fours, and the crossing over continues to happen. You look at the picture. More things start to happen. They're almost like pulling at each other's chromosome pieces or genetic material. So anaphase, same thing. The chromosomes are going to get pulled apart. Here's metaphase, here's anaphase. But this is the phase where the chromosomes start to get cut in half, okay? Now, what else do you see happening during this phase? Look at the colors of the chromosomes. The exchange of genetic materials happening, okay? So that's where you start to get variety. Think of you compared to your parents. You're a mixture of both of them, but also even the combination of that you got from one parent is a little off because their genes were kind of mixing. So you see that. Like telophase 1, similar to mitosis, telophase, where what happens? The cell cleaves. What else? Tell me everything that happens during telophase. The nucleus reforms. What else? Spindle disappears, and one more thing. The chromosomes become less visible. One more thing. Centrioles move back together. They end up in the same little spot, okay? Instead of apart, they come together, okay? Then meiosis 2 starts. So instead of cytokinesis, you have telophase 1, and then you have cytokinesis. Add that word there. Instead of interphase, starting up again, you start to go through meiosis 2. Okay? And then you start with 2 cells, and you end with what? 4. So if you look here, here's my 2 cells that resulted from meiosis 1, and they're going to go through the exact same thing that mitosis does. Prophase, metaphase, anaphase, telophase, exactly like mitosis. Look, chromosomes line up in the middle. They don't line up in fours. Crossing over doesn't happen. They don't line up with their homologous partners. Everything is the same. The difference happened in meiosis 1, okay? All right, now, and again, it ends with cytokinesis. So if you're thinking of the cell cycle from meiosis, it's interphase, meiosis 1, cytokinesis, meiosis 2, cytokinesis. If you want to write that down, feel free. Again, I said interphase, and I'm just going to put letters. I could get this to move. Cell cycle would be interphase, meiosis 1, cytokinesis, and then it leads to meiosis 2, cytokinesis. Yes? What do we call this red thing right here? Say it all together. Chromosomes. If you're not knowing these, you're way behind, okay, for studying. What do we call this thing right here? Centriole, okay? What do we call these things? Spindle. What do you call this dot right here? Centromere. What do you call this pattern right here? Tetrad. Or chromosomes lining up with their homologous partners. What do we call this right here? This right here. Crossing over, okay? Let me open this and see. What do we call this blue thing? Nuclear membrane. And everything inside is the, well, chromatin, but it's inside what? Nucleus. And if there's a dot right here, it's the nucleolus, okay? Don't forget these things because you have to know. All these things that I labeled here, you have to know. And actually, if you want to go back to those notes, right here, I labeled the nuclear envelope. Go ahead and label the nucleus. So you could remember. that's in there. And we'll add a dot, even though it's disappearing, and we'll add the nucleolus. Okay? Done.